So thank you very much for the opportunity to speak here today. I have a train to catch, so I'd better be on time. Let's put it like that. And I know it's a Friday afternoon, and hopefully it will be worth staying. And I should also thank Dana, because I think she just gave some of the introduction and motivation to what I'm going to talk about today. So as you might have realized by now, I'm going to talk about cells, single cells, but in particular the fact that cells are the basic building block of biology. It's not DNA, and it's not genes, and it's not molecules. The basic building block of life, which is irreducible, is the cell. Everything else cannot on its own constitute life. And in particular, in a multicellular organism such as ourselves, cells come in many shapes and forms and functionalities that people classify into skin cells and immune cells and different types of brain cells and so on. And they do it based on different types of characteristics, historically based on function and shape and morphology that you can see, and more recently based on molecular information, increasingly so. Now, as I think Dana actually pointed in her first slide, cells are also where genotype, the first place where genotype starts converting itself into a phenotype. And even though, to a first level of approximation, each cell in the body of multicellular organisms such as us has pretty much the same DNA sequence. Immune cells have some exceptions, and of course mutations also accrue somatically. But to a first level of approximation, this would be true. The genetic variants that exist between organisms manifest their effect in the action in individual cells. So a mutation in a gene that's important to the response in pathogens might manifest in the way that, say, an immune dendritic cell acts and would end up in a disease like inflammatory bowel disease or ulcerative colitis. Whereas a mutation in a gene that's important to muscle function would manifest in the way that skeletal muscle functions, even though the same mutation is present in the DNA of many other cells, and you would end up in muscular dystrophy. So to understand the genetic variation between individuals, we need to know the cells in which mutations act. It might be in a particular cell type. It might actually have multiple roles in different cells and in the interactions between them. So wouldn't it be wonderful if we had a handy atlas that would tell us everything we need to know about the cells in our body or in the body of any other multicellular organism? At the first level of approximation, we would want to know the overall types. For example, a neuron cell, which is what we see on the top, versus an immune cell. But of course, every cell can also reside in multiple states, and I think Dana gave a beautiful introduction to that. For example, an immune cell, when it is naive and has not seen a pathogen, is very different from an immune cell that has just encountered a gram-negative bacteria or its component, LPS, which is the one that I just animated in. Now, cells are located in a three-dimensional space. Even immune cells that can go through the blood and lymphatic system and enter into tissues, most of the time reside in three-dimensional space, and the vast majority of the rest of the cells are fixed in their location in a mature organism. And this location matters a lot to their function. Cells undergo dynamic transitions, and those can be incredibly transient, as we have just seen in the example of B-cell and T-cell development, or they can reside in a transient state for a longer period of time, and sometimes in the lab, we can keep them sort of in something that's like suspended animation, which is what we have for embryonic stem cells, for example. It's a very transient state in normal development, but we can make it perpetuate in a dish. And then finally, all cells in a multicellular organism, such as us, start from a single common progenitor cell, in our case, the fertilized egg or the zygote, and they are associated to each other through lineage relations that can often, but not always, be approximated by a tree. There are all sorts of interesting lateral transitions and things that can go back in time as well. And wouldn't it be wonderful if we knew that piece of information as well? Now, it's not that we're entirely ignorant. We know a lot, but we know also very, very little. And the knowledge that we have, nobody would argue with the fact that it's partial, and some of it is decades and even centuries old. And the older it is, the more reliable it might be, but it also highlights the fact that there's a lot that we might be missing. So I think this has been a preoccupation of biology for decades and centuries, and people have always been working at this problem. But in the last few years, technology is really opening up new vistas into this cell atlas, and it makes reconsidering the ambition of charting all cells in the human bodies with a new sense of urgency and opportunity. There are different technologies at the single cell level. We've just heard about protein level analysis. We've heard earlier about DNA level analysis. And I want to emphasize the unbiased view that is offered by RNA level analysis, because in the last few years, core technologies have emerged in the molecular biology level that would allow us to actually sequence with quite a high degree of reliability the RNA of single cells. There are ways in which to do sample prep, and I'll come back to this, that might scale to the right scale and precision and automation that lets us go after larger and ever larger numbers of single cells. And finally, there is an emerging computational toolbox from the work of many different labs, and we've heard an extensive set of tools in Dana's talk that goes from everything from classifying cell types to finding regulatory relations to identifying cell markers that allow us to prosecute things later on, again in the lab and in model organisms, and even tracing transitions and trajectories. These techniques are actually adaptable. People can grab them and run with them in individual labs, which means that you can rely on domain-level expertise in different biological systems in order to go after different parts of the atlas. This is not necessarily something that would be easy to do just by a single activity. You need real expertise. And in the case of RNA-seq, I like to illustrate it with the following numbers, just coming from the Broad, where I am, where the first single-cell RNA-seq experiment that we did, which was done in my lab, looked at 18 cells from dendritic cells, which are immune cell type from the mouse. And by March 2015, we were around 200,000 cells and counting. And actually, this number is hard to vet right now because there's no place where people say, I've sequenced a cell. So it's a guesstimate, and it might be an under-guesstimate. So let me show you, let me show you for a moment a bird's eye view of why an atlas is possible. And then for the rest of the talk, I'm going to delve into specific examples and tell you specific stories about work that we actually have done that will hopefully convince you that these are not just cartoons on slide. The first is, as Dan actually highlighted, is that to go after these problems, and we look at some specific numbers in a couple of minutes, we need a very large scale. So in work in the last few years, together with the McCarroll and the White's lab at Harvard, we have been working to scale up single-cell RNA-seq to the scale of tens of thousands, hundreds of thousands, and hopefully soon millions of cells to make sample prep at least as cheap as is possible. Sequencing is really beyond our control. And then applying these technologies can really let you go through the different aspects of a cell atlas. So starting with the classical question of distinct cell types, we can show that by measuring just a few hundred cells from the blood that are sorted to be specific dendritic cells, we can clearly distinguish the four known subtypes of dendritic cells that are known in the human blood. We can also identify, based on distinct transcriptional profile, a new unique subpopulation, and which comprises only 0.06% of peripheral blood mononuclear cells. And based on the genes that are expressed in those cells, we can predict that this might be a new lineage precursor that circulates in the blood. And this is a new finding for human. It exists for mouse. These are immune cells. They're considered to be a particularly convenient place to get started, partly because they don't reside in a tissue. But as I would show you, these types of approaches also scale to complex tissues. And here we're looking at 50,000 now retinal cells from the mouse retina, one of the most complex tissues that is out there, and where we can automatically discover dozens of subtypes, recapitulating decades of work in retinal biology in a few days of experiments. We can go after cell states within a single cell type. This is an example from Th17 cell biology, where we use about 2,000 cells, both from in vivo lesions mimicking autoimmune disease such as MS, and from in vitro differentiation, in order to all of which are Th17 cells par excellence, but they are in different states of pathogenicity, some being more pathogenic and some less pathogenic for promoting autoimmune disease, and that allows us also to find regulatory systems that control pathogenicity specifically rather than just the Th17 cell states. We can go further in cell states and look at disease, and not just at normal samples, and we can look at human disease, working with samples that come fresh out of the OR, in this case in the disease glioblastoma. And again, working on the scale of about 2,000 glioma cells coming from resected tumors, we can look at different aspects of cell state, all of which exist in glioblastoma cells. So the cell type is the same, but the state varies. We can start positioning cells in space. This is from the zebrafish embryo, where based on an analysis of 850 single-cell profiles coming from early embryo, and a tiny bit of in situ information with more traditional approaches, we can take hundreds of cells and put them at the right place in space. We can trace cell transition, transient ones, and that might impact a very tiny number of cells for a limited amount of time. This is from an ex vivo experiment using mouse dendritic cells, again, on the scale of 2,000 cells overall. Along a time course, we can identify a population that's present at about 1% and can be seen only at 1 or 2 time points, the short time window of 1 hour after the stimulation, which are early responders that are essential in order to coordinate the entire response. We can look at other types of transitions. This is an example from the cell cycle. By now, we've seen this phenomena in 2 different cell lines and in progenitor cells from the hematopoietic system. In all cases, you can look at unsynchronized data, and you've seen many beautiful examples of that from Dana today, and trace different types of trajectory and infer both the temporal order and the transient state of cells as they change over time here in the classical circular pattern. Overall, this shows that we can touch on almost every aspect that we would want to touch in an atlas. So that immediately grows your ambition and says, okay, how can one go after this in an orderly way? And are these just beautiful cartoons on a PowerPoint slide, or are they a real promise? So today what I'm going to do is that I'm going to touch on 3 aspects: the issue of type, state, and location, as well as talk a little bit about some foundational technology that we feel is going to be very useful for this type of cell atlasing. And mostly because he's actually here. This is Rahul Satija. He's sitting over there. He used to be a postdoc in my lab. He's no longer a postdoc in my lab. He's an assistant professor at NYU and the New York Genome Center. And his name is going to crop up again and again during the talk. And I wanted to make sure that if you want to hunt him down in the coffee break, you can. OK. So after the shameless plug for my former postdoc, let me start with technology. And actually, this is going to be a problem of scale. How do we actually get to the right scale in order to solve the right type of biological problem? If we want to actually build an atlas of the cells in our body, we better know how many there are. So first of all, the number is very big. There are on the scale of 10 to the 13 cells in our body, and this is after you let go of all the red blood cells that no longer have a nucleus. So fortunately, we don't have to worry about them directly. As Dana already mentioned, the classical number you will find on Wikipedia is 200 to 300 cell types. That immediately makes, indeed, the immunologists laugh heartily, because there are 350 or so clearly distinguishable immune cell types that are known to have distinct physiology. And worse than that, if you just look at the retina, there are about 100 subtypes that are quite clearly postulated or quite well known, again, for distinct physiology. So it's a large problem. However, it is finite. And finite is good. And it is finite in 2 senses. First of all, the number of cells is finite in any given individual. But more than that, you don't really have to measure each one in order to know what's out there. You just need to sample it at the right number in order to characterize the complexity that exists. So, for example, in the retina, in an adult human retina, there are about 150 million neurons. But given what we know on the subtypes that exist and the proportion of the rarest subtype, we can make simple statistical calculation on how many should we actually be sampling in order to recover the rarest subtype at a particular proportion. That would not take care of every transient transition that might exist only for half a day during human development, but it's a pretty good place to start. And that takes us down from 150 million cells to 50,000 cells, which is something that we can start imagining. So we have some idea of the numbers involved. Now, what does the technology, or at least the technology of a couple of years ago, was able to do? So we can look at the number of cells that can be measured with different techniques and the number of genes that we would be able to measure with those techniques. And I think this is a relatively safe assumption that most of the techniques work at the scale of 100 or hundreds of cells. And you could use some nice robots and liquid handlers and this and that and the other in order to maybe grow yourself in order of magnitude, but the cost still remains a pretty high cost of around $10 a cell that somebody just mentioned. I think Michael Schatz mentioned earlier for DNA as well. We would rather be somewhere around here where we can get, ourselves, a good 100,000 cells per experiment and hopefully drop the cost 1 or 2 or 3 orders of magnitude. So sample prep, at least, is no longer a problem. So in the first story that I'll tell you, this is what we attempted to do. And this has been a nice and very nice collaboration with Steve McCarroll's lab at HMS and with Dave Weitz's lab at Harvard in physics and chemistry. And this project was led by two postdocs, Evan McCosco in Steve McCarroll's lab, who's responsible to all the molecular biology, and Nonu Basu, a condensed matter physicist working with me and Dave Weitz. So what we decided to do is to try and go away from using plates or a standard microfluidics device, and instead of keeping cells in fixed registered position, keeping cells in something that looks like reverse salad dressing, which is basically droplets of water within an oil medium. And each of these droplets can hold itself a cell and a barcode, and might be able to capture the RNA in the cell onto the barcode so that we can then process it for sequencing, but know where it comes from. And this is exactly what we did in a protocol that we now call DROP-seq that works in the following way. We start with a complex piece of tissue. We dissociate it into a single cell suspension. We encapsulate those cells into these droplets, and each cell is encapsulated together with a distinctly barcoded bead. We then lyse the cells while they are still in the droplets, such that the RNA gets captured and barcoded on the beads, at which point we no longer need our droplets, and we can maintain these beads for as long as we want. And when we actually want to sequence a certain portion of them, we can select the number of beads that we're interested in, make a single RNA-seq library for 10 to the 4 to 10 to the 5 individual transcriptomes. This is a user-defined parameter of how many you can afford to sequence, and ship them off for sequencing. So I'm going to show you a few details under the hood. The first is the device that we actually use to do this. In this device, the cells come in a water medium, whereas the barcoded beads come in with a lysis buffer, and all of them meet the oil and get in a way that pinches them off and co-encapsulates the bead, the lysis buffer, and the cell, such that the cell will basically burst open and release its RNA to be captured on the bead. And you could have seen the movie running there. This looks like it's actually a very simple device. It is easy to make, but it wasn't that easy to get a device that would actually work. And, oh, now we missed the slide. These are the 37 designs that preceded the one that actually worked and took a couple of years to actually reach. So you need to have some stamina, and each device actually required Evan to do a whole series of molecular biology optimizations and testing. The other critical component is that we need these beads with the barcodes. So let's spend a moment on them. In each of these beads, we're going to have a cell barcode that is going to mark all the RNA from one cell with a single barcode. We're going to have this barcode in many, many, many identical copies on a single bead, and every bead is going to be different from every other bead. In addition, we're going to have what people call unique molecular identifiers, which allow us within a cell to mark each mRNA molecule with a unique identifier all its own. So now on this piece is going to be different between every copy on the same bead, and in addition, oligo-DT in order to capture mRNA. The most tricky part in this is getting these cell barcodes to work, because we will need about 10 to the 7 beads so that we can encapsulate with a Poisson loading only at most one bead and one cell together. So we will need 10 to the 7 beads, and each of the beads has to have many copies of something that's different from all the other beads. So we can't just sit there and synthesize it by design. We have to somehow make it very cheaply in a pool, and for this we use a trick that is called split pool synthesis, where we start off with a population of beads, and we separate it just by volume to 4 equal tubes or columns. And we do one round of synthesis, adding just one letter, A, G, C, or T. Then we take all of the beads from the 4 tubes, and we combine them together. So we now basically have 4 different types of beads, right, based on the letter that we added. We split them again, and we add another letter, A, G, C, or T. So after these 2 rounds, we now have 16, and we can continue splitting and pulling and splitting and pulling. And if we run it, for example, for 12 synthesis runs, we will get 16 million beads, each of them with a distinct barcode, or with an extremely low probability of having the same barcode on 2 different beads. So based on this trick and some additional synthesis steps, we end up with our barcoded beads, and now we're ready to see whether all of this was a nice cartoon or actually works. And in order to do that, we use this very simple design of taking cells from 2 different organisms, mouse and human, pulling them together, running them through the device, and seeing what happens. And we're going to plot our results in the following graph, where on the x-axis, we're going to quantify for every barcode, for every bead barcode, every cell barcode, we're going to quantify the number of human transcripts that are associated with it versus the number of mouse transcripts. If the results are good, then maybe it came from a single human cell and should lie close to this axis, or it came from a single mouse cell and should lie there. If the results are not so good and the cells are mixing together despite all of our attempts, it's going to lie somewhere here. But there's actually a much worse result, which is we can't even get RNA-seq to work, and it's lying down here. And this is where we started. Okay? So this is a story of optimism, because it's true that this is where most of them started, but there were things like this, which, given the number of points that are here, are still remarkably unlikely to have occurred completely by chance. So we said, or rather, Evan and Ani said, there's hope, and there's better hope, and better hope. And after 18 months of relentless work and 37 devices and many different bead designs, you end up with something like this, where once in a while you get a cell mixing, and most of the things look incredibly clean. And this is where we rely today with extreme precision and over 95% cell of origin specificity. We can get this as high as 99% if we run the devices slower and slower. We also capture a good number of thousands of distinct human genes, which is a good performance compared to state of the art. And we capture about 12% of the single-cell mRNA, which is extremely comparable to state of the art. So now one can ask oneself, what can we learn from just 10% of the mRNA content of a single cell? And in order to do that, the first thing that we said is, well, we were running all these experiments with two cell types, human 293 cells and mouse 3T3 cells. These, for example, 293 cells, here are 569 of them from one of the QC experiments that I just showed you. They're cycling cells. They're undergoing the cell cycle. Can we distinguish the cell cycle? So we went in with two signatures, one for G1S phase and one for G2M phase, based on historical microarray studies from about 10 years ago. And we got this beautiful ellipsoid trajectory, which is lost. Everything fills in when we shuffle the data. We can go and take the cells, both from human and from mouse, and score them for each of the phases of the cell cycle. And we get an almost continuous sampling of the cell cycle, suggesting that we can actually identify very nicely cell states. And we can now score each gene, and we can find hundreds of new cycling genes. Now, if you're as old as I, and remember the historical papers on microarrays saying that human and mouse don't have conserved gene expression programs in the cell cycle, it's probably people couldn't synchronize them in the same way. Because in asynchronous cultures, they actually have perfectly conservation of the cell cycle. OK, so we have a technology at hand, and we can find some cell states. Can we now take it to the next level and start looking for complex cell types? So let me first just conclude what we have in our hands. Dropseq is scalable. We can go 10 to the 4, 10 to the 5 cells per experiment. It's flexible. We can choose how many cells we want to sequence once we capture the RNA on the beads. It's sensitive, it's specific, and it's accessible. It doesn't require any type of expensive equipment in order to run. And you can run a lot of these cells per day, and it costs you about 6 pennies per cell to prepare your RNA-seq libraries, compared to the $10 that it costs us in any other technology. We can find cell cycle states, and now we asked ourselves, could we use it to define cell types? So this is the first scale of our atlas, right? Let's find different cell types. We weren't even there yet. And to do that, we turn to the mouse retina, which, as I already pointed out, is one of the more complex pieces of tissue that is known, and where cell types are actually characterized already. So we have some extensive textbook knowledge to fall on. And I'll give you a very crash introduction into the retina. We start with the retinal pigmented epithelium, which is actually not neurons. In it, there are two types of photoreceptors that take the light signal and transduce it into neural signal, the cones, of which there are two subtypes, and the rods. These are connected through a series of connections, eventually to cells called retinal ganglion cells, of which there are 30 types, that will send the signal back to the brain. The signal gets processed by a set of interneurons, which are neurons that control neurons. You have horizontal cells that connect cones and rods. You have bipolar cells that connect cones and rods to retinal ganglion cells, and you have the Muller, and you have Amacrine cells that connect the retinal ganglion cells to each other. And in addition, you have glia and some additional non-neural cell types. The rods make 80% of the retina. So by definition, all of these other subtypes, the 30 subtypes of retinal ganglion cells and so on, they're all going to be very rare. So it's a difficult problem to solve, but we know a lot of the answer. Plus, we had the secret weapon, which is Joshua Sainz, one of the best people in the grand priest of the retina, collaborated with us. That was very helpful. So in a series of 7 biological replicates, Evan ran 44,808 single cells. The vast majority of those cells were run over a series of 4 days, so this was a pretty rapid data collection effort. We selected the best 14,000 cells, those that had the most transcripts in them. We then selected variable genes out of them, identified 32 significant principal components. Then, and this saved me a lot of work that Dana talked right before me, we used TSNet that Dana described to you in order to generate a nonlinear embedding of the 14,000 cells. We projected the remaining 75% of the cells into this map. We used density clustering in a standard approach because this is not as complicated as the data Dana described to you in order to identify clusters. And we then, in post hoc, merged some of the clusters that did not have support from differentially expressed genes. And all of this work was done by Rahul, that I already showed you his picture. And this is what we got. This is definitely a pretty picture, and it's colored by automatic clustering. No human knowledge of the retina was imposed up to this point, but the question was, of course, if we compare it to known retinal subtypes, will there be a good correspondence between the information? So Rahul took this data, organized it into a dendrogram. They printed a piece of paper. Evan and Rahul went down to Harvard Square, sat in Josh Sainz's office, and started annotating based on what is needed. And this was in slightly older data. There were less cells involved, but with the full data set and with some nice, with the help of some nice graphic editing, this is what we end up with. So here are 30, whoa, I don't know why I wrote here 34. Here are 39 clusters, of which 33 are neural cells, and the remainder are things like Muller glia, astrocytes, fibroblasts, and so on. And they're colored now by their retina color code that I showed you before. And you can see that we find cluster upon cluster of amacrine cells. We distinguish the rod from the cones. We distinguish the bipolar cells from the amacrines, and so on. So we can capture very well many of the different subtypes. We see here also cell expression markers that are known to characterize the different subtypes, and they fit the classification really well. The proportions also fit quite well. So here we're comparing the proportion based on the drop-seq estimates versus the proportion based on the literature review of the retina, and things fit quite well. In some cases, like the amacrine cells and the bipolar cells, we're capturing 50% of the subtypes, of the number of subtypes that is expected in a cell type, which is quite remarkable because we only ran 50,000 cells and 80% of them come from a single type. That's the rods. And in another case of the retinal ganglion cells, where we know that there will be over 30 subtypes, we actually can separate them further because they are so rare. They're less than 0.1% of the retina. So we simply haven't sampled a sufficient number of cells. We will have to enrich for them. Now you can zoom in. This is the case of the amacrine cells, of which we have the largest number of separations. And you can see that you can go into further classifications that are still correct. So we're distinguishing the GABAergic from the glissinergic, from the excitatory, from the starburst amacrine cells. These are all distinctions that are textbook distinctions in retinal biology, but we can also see that these known markers cannot account for all of the sub-separations that we find. So we can now go for each of the clusters and attempt to identify markers that classify that cluster. For example, for cluster 20, we have triple P1R17 that appears to be a very distinctive marker specific for this cluster. And Josh's lab can go back into the retina and actually demonstrate that this is a distinctive cell type. And that can be associated with specific anatomical and physiological features. And finally, just to tie this particular loose end, if you remember, I started by telling you the genes that are variable from genome-wide association studies, will have to be understood within the context of the cells that they operate. So as an exercise in this idea, we went back to the first successful human GWAS. That is the study for acute macular degeneration, which is a disease of retinal degeneration. We identified 11 of the 18 mouse orthologs of genes from that classical GWAS study. And we can show the cells, which are mostly actually not neural cells, in which these genes are actually acting inside the retina, which is probably where the disease arises. And this is actually consistent with the known disease biology for AMD. So to conclude this second part, I showed you the drop-seq can be successfully applied to complex tissue, that we analyzed almost 50,000 cells across 7 replicates. I didn't show you, but the results are extremely reproducible from one replica to the next. Most of the cells were collected during only 4 days of experiment. We have a robust and sensitive computational strategy to identify subtypes in this system. We found 34 retinal subtypes from a whole tissue experiment. Many of these conform to decades of research in retina biology. But we also identify new cell type markers and new relationships, and we can infer cell of origin in disease. And it is important to know, though, that the very rare subtypes, for example, the RGCs, will require some specific enrichments. Unless you really want to blow all your sequencing on rods, you'd better just look at retinal ganglial cells and then dig deeper into them. And that's going to be, I think, one of the strategy of atlasing for types. OK. So I'm pretty good on time, so we can move on nicely into the second part. So we looked at cell types. Can we now think a little bit about cell states? And for this, I'm actually going to transition into the context of disease. Complex tissues exist, of course, in the context of normal tissues, such as the retina. But we can also look at other types of complex phenomena in human biology, and in particular in the context of disease. And for this, looking at RNA-seq has several types of benefits. The first are all the things that I just showed you. You can look at how expression looks and find markers and understand biology and talk about the cell cycle, and we'll see all of those things. But the second is that RNA is actually sequence, and that sequence comes from the DNA, which means that the RNA carries with it something that we fondly call the expressed exome. It's a window into the epigenetic variation, but it is also a window, a limited one, but still one, into the genetic variation between cells. So it might allow us to identify SNPs or splice isoforms or cases of gene fusions, and maybe look at the associated microbiome and virome of a tissue, and so on. So it really tells us something about genetic material as well. So this part of the story, the cast of characters, is changing a little bit. This was a collaboration with Mario Suva and Brad Bernstein and David Lewis's group at the MGH. Working with 2 neurosurgeons, Anup Patel and Andrew Venteicher, and a computational biologist in my group, Itai Tirosh. So the disease that we're going to look at is glioblastoma. Glioblastoma is a terrible disease. All diseases are terrible, but glioblastoma has its special role. It's the most frequent human brain tumor. There aren't that many cases diagnosed per year, but it is the most common adult brain tumor. And median survival is 15 months. 100 years ago, survival was 6 months. The difference is because diagnosis is earlier. There's really nothing that can be done. This is a really bad disease to get. And as you can see with this, well-invasive tumor in the brain. Glioblastoma is also interesting if you're thinking about cell diversity. It's called also glioblastoma multiforme because it has all of these faces. There is variation between different types of tumors. People call these subtypes, and it has prognostic value, and people can show that with gene expression profile. There is also variation within a tumor between the different cells, and in particular, people distinguish between cells that can propagate the tumor, especially as you transplant it into an animal, and cells that cannot and are already differentiated. So we thought it would be interesting to use single-cell RNA-seq in order to identify signatures of glioblastoma heterogeneity, and we set up the following pipeline, where we start with tumors that are coming from the OR and are resected at the OR. Then they are dissociated. In this initial study, we depleted all the immune cells with the CD45 depletion to obtain a suspension of presumably single tumor cells in lysis buffer as singles. And at that point, you can bank it or send it off to sequencing, do whole transcription amplification, and sequence. And the timeline between the tumor being resected in the OR to the cells being lysed as singles in the wells of a multi-well plate was 3 hours. We now are down to 2 on this process. And overall, we analyzed 430 cells, around 100 from each of 5 patients. And some tumor genetic details. The first question we asked ourselves are they tumor cells? Surgery is by no means perfect science, and this tumor in particular is very invasive, so there could be other cells lying around. And we said, oh, if we only had DNA, if these people were only consented for DNA sequencing, we would be able to do something like this, a CNV profile, and find amplifications and deletions that are characteristics of the tumor. And then we told ourselves, well, maybe we can actually get this information from the RNA, at least at some level of resolution, by sliding a window, something similar to what Michael described, but much, much simpler analytically, by sliding a window of about 100 genes or so, looking at the expression level within this window in the single cell, and comparing it to the average expression in this window in some typical brain tissue, in our case from the GTEx, with the idea that there are going to be a lot of differences at the level of any individual gene, but by the time the window is big enough, most of the signal that will remain should reflect copy number variation. This sounded like an idea that should not work, so we first tried it in the bulk, in a case when we knew the answer, which is the CCLE cell line. Here are the CNVs. Here are the CNVs as they are inferred based on SNP arrays and, I guess, GSTIC. And you can see that there are some large-scale aberrations, and then there's this punctate pattern of these little dots. These are small-scale aberrations. This is the inference based on the RNA-seq data from the exact same cells from the CCLE. And we can see that the RNA-seq nicely infers large CNVs. The correlation is quite well. It kind of misses out on all of these fine details. But this should be sufficient to tell us whether our single cells from the patient's tumors are singles or not. So we did exactly that. We took each of the single cells. We did this inference relative to GTEx brain. We used standard clustering, hierarchical agglomative clustering, completely blindly to which patients these samples came from, and we started looking at the patterns. The first thing that we saw was an amplification of chromosome 7 and a deletion of chromosome 10, which are the canonical CNVs of glioblastoma. The second thing that we saw is that this clustering basically clusters by patient, and that at this crude level of CNVs, the samples are mostly clonal. And we can also see that in some cases we see subclonality, and in this case even mutually exclusive subclones in terms of the CNVs. There's also this bunch of cells up here that look quiet. They don't seem to have CNVs. One of them is the GTEx brain. That's our control, so obviously it should be there. 9 we could identify as oligodendrocytes, and 1 monocyte. So we really depleted the immune cells from these samples quite effectively. But we know that we're now handling in tumor cells. So now we can go back to our question of what's epigenetically different rather than genetically different between these cells. And the first question we asked ourselves is what is going on within the tumors in terms of their stemness state. So for 3 of the patients, in parallel, Anup and Mario took the cells, derived them in culture to either differentiated differentiated glioblastoma cells in the presence of tumor, or in the presence of serum, or in the absence of serum, into stem-like cells, collected RNA-seq profiles, made a signature of the differentiated versus the stem, and then scored the cells coming straight out of the tumor that have never seen a dish but were lysed right away against this signature. And we found that they actually span the spectrum. Rather than having a special subpopulation of stem cells, what we actually see is a spectrum of cells, starting from ones that look the most differentiated to the ones that look the most stem-like. This is from one of the tumors, but the same phenomena was true for each of them. And this actually mimics what we see when we stain cells for antibodies for likely stemness factors, that you get this full rainbow of behaviors. We can use this kind of variation to our favor in order to look for dependencies between expression of genes and other genes in order to try and nominate factors. Some of the factors that we find, like POU3F2, are also found in vitro to reprogram cells into ones that can promote tumor in mice. But others, like NFI and NFIB, we see only in vivo, and they allow us to elaborate how these stem-like programs are controlled by these regulatory factors. Last, we look at the variation that is known to exist between tumors as they are analyzed in the bulk. And in particular, there are 4 different transcriptional types that were defined in glioblastoma: mesenchymal, preneural, classical, and neural. So we took each of our cells and we scored it against these classical signatures that were scored in the past. So now we have our 4 TCGA subtypes, the cells from each of the patients. And what we get again is a spectrum of states. So in each of the tumors, some of the cells come from a single dominant subtype. For example, in MGH30, the majority of the cells are classical. And if you looked at a bulk MGH30 tumor, you would call it a classical tumor. But there are always at least 2 minority subpopulations, in this case, preneural cells and mesenchymal cells. And in some cases, you even get cells that appear like they're transitioning between these different states because they have signatures of both, as we see in these hexagon plots, and that's true for all 5 tumors. And finally, this kind of heterogeneity can now be taken back to the bulk samples, and we can try to infer back from the bulk samples how much of these minority subpopulations might exist by testing whether the bulk sample profile is explained better, for example, by having only preneural-like cells, or having preneural-like cells and a low content of alternative subtypes, or a high content of alternative subtypes. And the bottom line is that the larger the heterogeneity in subtypes inside the tumor, and these are epigenetic subtypes, not genetic ones, the larger the heterogeneity in them, the worse off the prognosis. So to conclude this part, I showed you that single-cell genomics can be successfully performed on fresh tumor samples, that this is an end-to-end process. It requires a partnership between a neurosurgeon and a pathology lab and a molecular biologist and computational biologist. The transcriptome gives us a window into the expressed exome. I showed you CNVs, but to some extent one can also score SNPs, mutations that affect splicing, and so on. The tumor cells form a continuum from differentiated to stem-like cells, and single-cell data can help predict key regulators of the TPC state. In each tumor, we see a dominant transcriptional subtype and other subtypes, and the subtype heterogeneity has prognostic value. Now, in unbridled optimism, I think I had too many slides. This is something I took out and in and out and in. I'm going to skip to here, because I do want to finish on time. So these are single cells. We've seen single cells from the retina. We've seen single cells from the tumor. But tissues, they're not just a bag of cells. These cells are organized, and they're organized in very precise ways. In the case of the retina, this physical organization is critical for retinal function. In the case of the tumor, this physical organization can be critical for whether the tumor is more invasive or not, and to the relationship between the tumor and immune cells that infiltrate it, and so on. So wouldn't it be wonderful if we didn't just know what the cells are, but also where the cells are placed? So in the last part of the talk, I'm going to tell you a little bit about how we're starting to look into this problem, starting with canonical tissue that looks the same way every time, and using a computational strategy to infer the spatial position of individual cells. And this is work that was initiated and led by Rahul in his very last couple of months, or 3 months in the lab, in collaboration with Jeff Farrell, a postdoc in Alex Shearer's lab, and they gathered a research associate. So we want to solve the following problem. Imagine that there are 3 cells, A, B, and C. These cells actually reside in particular places in the tissue. Given single-cell profiles of A, B, and C, can we put them back into place? And the solution that we have devised for it is a method that Rahul called by the beautiful name Seurat, after the pointillist painter Seurat, to try and invoke the idea that each of the cells is like a pixel in a beautiful picture. So here's the Cliff Notes version of Seurat, and I'll drill a little bit in. The general idea is incredibly simple. We will have single-cell transcriptomes of cells. We can take a piece of tissue, dissociate it, get single-cell profiles. However, For a few of the transcripts, a small number, and we'll call them landmark transcripts, we're also going to go to traditional biological experiments and get ourselves some good old-fashioned in situ data. In our particular example, it's going to be colorogenic in situ, the sort of low-end, the low-tech version of in situs, but you can, of course, have finer versions as well. And we're going to use these landmarks in aggregate in order to tell us where cells came from. So now I'm going to drill into some of the critical aspects of how we make this work. So first thing that we need are these spatial landmarks. We start with an image that starts like this. We see Jeff down in order to make a binary representation of it. What should it look like? That depends on the system that you study. The resolution that you should do, that depends on the system that you study and the data that you have at hand. In our case, each of these bins is going to be about 100 cells because that's the resolution at which the images and the biology actually takes place. And now we're going to have this kind of information for multiple different genes. For example, gene X, gene Y, and gene Z. Overall, this is going to make us a picture that looks something like this, right? Here is information on these 3 genes. Now we're going to take a cell. Here's a cell, for example, that expresses the red and the blue gene, but not the yellow one. So we can say, oh, it must have come from here. Here's a cell that expresses the yellow and the blue gene, but not the red one. So it must have come from here. That seems rather simple. That's the idea. Now come the details. The details do matter. One of the most critical details is the fact that, unfortunately, single-cell RNA-seq is far from a perfect method. It has a pretty big false negative problem. So stuff can be expressed, yet not detected in single-cell RNA-seq. So, for example, if I look at this cell that actually came from that place and should express red and blue but not yellow, oops, maybe in this particular profile, we missed blue. Now, we only have a few of these landmarks, right? If we could do an in situ for every gene, why bother with single-cell RNA-seq? So we only have a few of these landmarks, and if we missed a piece of information on blue, we would get the cell all wrong, and we will say it came from somewhere here, rather than from that very particular corner. So that seems like a pretty big problem, but biology does come to the rescue, and this is because genes are typically co-expressed with other genes. So if I have my landmark here and I can't really determine how it's expressed in my single-cell profile, I can take a peek across all single-cell profiles. And this gene is typically going to be co-expressed with many other transcripts. Now I'm just in my single-cell profiles. I don't have any spatial information on these transcripts, but I do know whether they're expressed or not. So I can impute a much cleaner version of the biology than the data that I actually measured by using these co-expression relationships. So now, if I actually know the gene P, Q, and R are all co-expressed with gene Y, they likely have the same spatial pattern, even though I don't know that spatial pattern. I don't have that data in hand. I only have their single-cell profile. But given their single-cell profiles showing that they're all expressed, I can infer with a nice linear regularized model that gene Y is expressed in this cell as well, and all is well again. Everyone goes back to its place. So this is probably the biggest problem, and it is quite solvable. So now, all the way from the top, I still have a few minutes, we have single-cell RNA-seq profiles. We get expression data. It's noisy. We handle the noise with imputation. We have a spatial reference map. It looks kind of shady like that. We binarize it, make a cruder map, but this map is going to be sufficient for us. We still have a couple of remaining problems. This data has dynamic range. That data is binary. We need to match them somehow. So we actually now take this data and the proportion of cells that we are expecting to express each of these genes and fit a mixture model. And so we now replace the original binary data with data that's fitting to this one. And all that remains to do is to calculate the probability of each, based on each gene of the cell mapping to a location, summing the long probabilities, and projecting where the cells are most likely to come from. So now for every cell and every bin, we have a probability that the cell came from that bin. So again, like many other points in the talk, this is a wonderful cartoon, but does it actually work? So to test this, we took the test case of the zebrafish early embryo, analyzed 851 cells from 50% epibole, in parallel, we took 37 landmark published profiles. Not a single experiment was done for in situs at this point. This comes from databases, mostly Ziffin, and that identified 37 landmarks that cover different aspects of patterning in the zebrafish embryo. And then we mapped the 851 cells into bins and got some result. But the question, of course, is this result correct? Do we get it right? So for this, we were fortunate that Jeff is a great experimentalist, and he can actually go, this is a mouth pipette, and he can actually pick cells. And he can actually pick cells and know where he picked them from within an error of plus minus 1 bin in every dimension. And he did this 28 times. So we have 28 cells that we know where they came from pretty precisely. And now we can map them without using this knowledge and asking whether we get them right. So here is one example. In pink is the presumed origin, according to Jeff. In purple is the probability that Sarah assigned to the single cell profile. And in green is the centroid for these probabilities. And you can see that we got it pretty close. This is the best. You can see the worst example out of 28. And on average, our median error is 2 bins, 1 in each axis, which is Jeff's own estimate of his visual acuity at actually telling us where that cell came from in the first place. So we were pretty happy. We were actually really happy. Now, if we actually are putting cells in the right place, every cell brings with it a whole gift. It brings a whole expression profile, not just the landmark genes. Those we knew we knew where they were expressed to begin with. So if this is a bunch of mapped cells, and this is some gene X, and this is the expression level of gene X in this purple shading, we can now take the expression level of the gene in every cell, weigh it by the probability that the cell is mapping to every bin, sum over all the cell, and infer ourselves an in situ map for this gene. And we can do it for any gene that we have expression for. So once we do it, we get a bunch of in silico in situs, I guess we can call them, right? These are just computations. We don't know if it's correct, but there's no reason to assume that it's not. So for 15 of these patterns, Jeff went and did in situs, and they match in all 15 cases. So now we can get a genome-wide in situ map without doing the in situs, and over time convince ourselves that these are actually correct. There are other tricks that we can play with this. For example, we can look for new cell types that have distinctive spatial patterns. So here is a way of finding a new population without any spatial information, just from single-cell RNA-seq data. You run the simplest PCA. You look at some PCs. You find something that seems to lie out, in this case, this purple population of cells. And you look at some gene expression. You see that they're expressing some apoptotic genes. So that's potentially interesting. Then you can ask yourself, how are they mapped, those cells? And they seem to be mapped in this pattern that we call salt and pepper. And salt and pepper is particularly terrible, right? Because there's nothing distinctive about this pattern, which means that when people saw this, and people saw this before for some of these genes, they thought it was stochastic. Some genes are up here, some genes are up there. They couldn't tell that they're actually a distinctive subpopulation, but our single-cell profiles suggest that they are. And they even allow us to find markers by comparing each of these cells not just to all the other cells, but specifically to the other cells around it in the same physical neighborhood that are not of the same type. And that's critical in systems like these that are organized with gradients. And now Jeff can go again and do in situs not just for one, but for pairs of genes, and show that these genes are indeed expressed in a salt and pepper pattern in a coordinated way across cells. This is not a stochastic pattern. This is a real new subpopulation, and Alex Shear's lab is now studying this subpopulation and its role in morphogenesis. So to conclude this last part of the talk, I showed you that we can combine single-cell RNA-seq and even legacy in situs out of databases. We didn't go after pretty in situs. We went after typical in situs for this study. We did it with the test case of the zebrafish embryo. We can correctly map the cells to their known location. We can correctly recover known and novel spatial expression patterns. We can successfully query for patterns of interest. I actually didn't show you how we do that. And we can successfully discover new cell types that have distinctive spatial expression patterns or distinctive spatial patterns. If you have a reference map to start with, that's great. But if you don't have a reference map to start with, there are ways around that problem too. For example, you can start with single-cell analysis, look for variable genes, make yourself a bunch of in situs, start mapping cells. As you map the cells, you will get a new bunch of in silico in situs, find new patterns, repeat. So you can kind of bootstrap yourself. There are also new spatial ways, which are not single-cell resolution, but new ways to look at spatial expression patterns that can help you collect this on a genomic scale. It introduces interesting notions in power analysis. You can start thinking about the number and spatial diversity of required landmarks and your ability to resolve, so the power to resolve things for spatial patterns. And we think that this general idea of using one type of metadata, which might be very difficult to get, with another type of data in biology that might be easier to get, and use one in order to impose this information on the other, is probably going to be useful. And one can only think about, for example, antibodies are rare, but tell us a lot about proteins. RNA is easier to measure, but we might not be able to do as many cells, and there might be a match made in heaven. So to conclude, I hope I convinced you, and hopefully also with Dana's talk maybe even more so, that a human cell atlas is within reach. It will require some discipline. It will require some scaling of methods. It will require a lot of computational methods that will be scalable and sensitive and robust and have all of the wonderful features that they need to have. And it will require a partnership across biology, because unlike a lot of other problems, cells are not like nucleic acids. They're very special. They're finicky. They have their own traits. You need to know how to handle them. But having said that, one can go after their types, as we have done for the retina. After their states, I showed you in gliomas. I kind of touched very quickly on pathogenicity in T cells. You can look for their location. You can monitor transitions. And you can even look for lineages. And with this, I will conclude. I just want to repeat the thank yous for the many collaborators and people who did this work. The work on Dropsic and the retina is a collaboration with Steve McCarroll's lab, Dave Weitz's lab, and on the retina with Joshua Sains's lab, and was really led by this humongous effort by these two highly optimistic postdocs, Evan McCosko and Oni Basu, and Rahul played a critical role in the analysis of this data. The work on glioblastoma was a partnership with the Suva and Brad Bernstein's lab, and this is still ongoing, especially Anup Patel, a neurosurgeon, and Itai Tirosh, a computational biologist. And the work on Sarah was led and initiated by Rahul, and was a wonderful collaboration with Alex Shear's lab, especially with Jeff Farrell. There were multiple sources of funding. Since I'm here, I really want to highlight this. Dropsic was funded by one of these seed grants that Simon Center at MIT, for the social brain at MIT grants, and I don't think we could have done the 37 designs and all the MySix that went with them otherwise. And with this, I will finish.